much genetic material here as it was here, but the genes are all the same. We have the same genes that are coded for here that are coded for over here. So chromosome numbers remain the same. So after which phase do cells become haploid?
Telophase.
After telophase 1.
So there's 2 chromosomes in telophase 2 and only, and then there's 2 in after telophase 2.
Exactly right.
But
You know,
between the 2 cells, right? So each of them only has
each of them has 2, right? Each has 1, 2, 1, 2.
Okay.
So let's think about this again, comparing mitosis and meiosis. Can I erase this? Is that okay? So in mitosis, let's say we have a cell that has, we'll say it has 6 chromosomes. Get crazy. Six chromosomes, right? What's the purpose of mitosis?
To divide.
To create 2 daughter cells that are genetically identical to the mother cell, right? So we want to have 2 cells that come from this. Now we know that before mitosis can occur, the G1, the S, and the G2 phases have to occur, right? And it's in the S phase that all of our DNA is synthesized and duplicated, right? That's how we get chromosomes that look like an X. So if we want to put 6 chromosomes in each one of these cells, what we're going to do is we're going to take those chromosomes and separate them, right? And so half of this chromosome is going to go into here, and half of it's going to go over there. And that's cool because those 2 halves, those sister chromatids, carry the same genetic material. They're just duplicates of each other. And so in the end, this is going to look something like this. Right? It's identical to this one. Has exactly the same genes. Same with this one over here. We're going to get 3 of these, and we're going to have 3 of these. That's mitosis. Meiosis is a little bit different though, right? So in meiosis, what we do is we start off with this same cell that has our 6 chromosomes. That, right? So if these are homologous pairs, during prophase 1, those things start to align, right? In metaphase 1, these homologous pairs are all lined up exactly like that at the center of the cell. So after meiosis 1, we're going to have 2 daughter cells. And the question is, what do those nuclei look like, right? How many orange chromosomes is this cell going to have?
3.
It's close to 3.
2, 6.
So it's after this phase, right, of meiosis 1 that we end up with 2 haploid nuclei, right? This is the stage in which it becomes haploid. So these chromosomes have to segregate from each other. So what's going to happen is these 3 on the right are going to migrate down here into this cell. And these ones on the left are going to migrate down into this cell. Right? And so we're going to have 2 orange and 1 blue over here. Does that make sense? So we'll have something that looks like this. You okay with that idea? There's some nods, but I want to make sure everybody's nodding. Okay. And so over here, how many blue are we going to have?
2.
2. Exactly right. And then 1 orange, right? Okay, so how many chromosomes does that cell have? Do each of those cells have? 3, and we started off with 6. So it's at this point, after telophase 1, they become haploid. Okay? So then we know that we're going to go through another nuclear division. So each of these cells is going to produce 2 daughter cells. Right? Okay. Question is, how does that look? How many blue chromosomes is this cell going to end up with? It's close to 1. It's 2, right? Because what happens is sister chromatids segregate and go into different cells. So this sister chromatid is going to come down here, and this one's going to go over here. Same thing with this one right here. This sister chromatid is going to migrate down into that cell, and this one's going to migrate over here to this cell. So we should end up with something that looks like this. You okay with that? So how many orange ones is this going to end up with? 2, right? Because sister chromatids from those 2 orange chromosomes are migrating into different cells. So we're going to end up with 2 orange here, 2 orange there, and then a blue apiece. So do you guys see that this division is almost identical to this division? Right? We started off with 6 chromosomes here, and each of those daughter cells got a sister chromatid. Same thing happened here. We started off with 3 chromosomes, and each of those daughter cells got a sister chromatid. So we retained the number of chromosomes between this division and this division. Carly, do you have a question?
I just want to clarify something so I can make sure I'm that's right. So then during telophase 1, when the crossover occurs, is that why when we get down to if I can use my words right.
Meiosis 2?
Prophase 1. So during prophase
Okay.
1, when the crossover occurs, is that why when we get down to telophase 2, they're all unique because they have different crossover sections?
Exactly right. So each of the chromosomes should be unique, and each cell should have a unique composition of chromosomes. And we'll talk about those ideas in just a few minutes. But from a macroscopic level, does this kind of make sense, the difference between mitosis and meiosis? Okay, Linda.
So at the very bottom, when it replicates into 2 more,
Uh-huh.
each of those sister chromatids, each of those have to have the same amount in there? Like one would have 4 and one would have 2?
Right,
It
and
has to
so,
be exactly the same?
and we're going to talk about that chromosome abnormality a little bit later.
Okay.
But when non-disjunction events occur, like trisomy 23
Yeah.
or trisomy 21, this is where they're occurring. One of these cells could end up with an extra chromosome,
Oh.
and that causes things like Down syndrome.
Okay.
Yeah. Other questions? That was a good one. Okay. Would you guys be able to replicate a diagram like that on the exam?
Yes.
Okay, yeah.
So like,
Sophia.
on the first one, is that like,
The first one right here, mitosis.
yeah, so,
Okay.
no, meiosis.
Okay, first one right here.
Yeah, so is that like meiosis 1, meiosis like,
Yeah, do you want me to write that?
yeah.
Okay. So I'm going to put meiosis 1 here, but understand that this is, I'll put it right here.
Yeah,
1, and this is
okay.
meiosis 2, and then this is okay, so why do we even care about this? Why is this a cool thing? This is a cool thing, you guys, because this allows us, meiosis allows us to retain chromosome number across generations, right? Think about what would happen if our gametes looked like this. If each of our gametes carry 6 chromosomes, if this 6 chromosome sperm cell fertilizes this 6 chromosome egg cell, we end up with 12 chromosomes, which is bad because the parental generation only had 6 chromosomes. So we've effectively doubled the number of chromosomes, right? But if we go through meiosis, and this is a sperm cell and this is an egg cell, we get a sperm cell with 3 chromosomes that fertilizes an egg cell with 3 chromosomes, and 3 plus 3 is 6, which is the same number as the parental generation. Meiosis allows us to retain that number of chromosomes across generations. Okay? So, I said it last time a number of times, but it's really hard to understate the importance of genetic variability in the context of evolution, and sexual reproduction is a primary way in which we introduce genetic variation. And that genetic variation isn't necessarily novel genes, but it's novel combinations of genes. So what we do with sexual reproduction is we compare different genes and different alleles in unique and hopefully wonderful ways. Think about crossing over. What we've done is we've taken the chromosome that carries eye color and a bunch of other genes from mom and paired it up with that from dad, right? And so maybe dad has green eyes and mom has brown eyes. And we've taken dad's green eye gene and stuck it onto mom's chromosome, and mom's brown eye gene and stuck it onto dad's green eye chromosome, right? So now all of a sudden, dad's green eyes are paired with a whole bunch of unique genes and alleles. So crossing over is a really, really important way for us to introduce novel combinations of genes. Additionally, it also really holds those prophase 1 chromosomes together really tightly. So when things are being pulled about by the mitotic spindle, they don't separate when they're not supposed to. Okay, so this is how meiosis 2 looks, right? So we start off with prophase 2, and those haploid cells their chromosomes begin to condense, and the nuclear envelope falls apart if it has formed. In metaphase 2, those homologous chromosomes, the only chromosomes that are left in cells, are aligned on top of each other. And then in anaphase 2, sister chromatids are pulled apart. Telophase 2, new nuclei form around those chromosomes, and there are a haploid number of unitary sister chromatids. And we end up with 4 cells that are all haploid and genetically unique. Okay. Questions before we talk about some relatively different things? Okay, so this is what crossing over looks like, right? We have a chromosome from dad and a chromosome from mom, and during prophase 1, they align. They're paired up and they're held together by cohesins. And then occasionally, and actually it's lots more frequently than occasionally, they'll exchange genetic material by flopping over on each other. This leg flops over on top of that leg, and they exchange genetic material. What is remarkable about this process is the exact same amount of genetic material is exchanged every time. That is, mom's chromosome doesn't end up with more of dad's than dad does of mom's. That's really important, because if you end up with duplicate genes, like if you've given up a little bit too much or not taken quite enough, that can cause all sorts of genetic problems. So this crossing over, for whatever reason, occurs so that there is the same amount of genetic material exchanged between homologous chromosomes. Okay, so let's talk a little bit more about the differences between mitosis and meiosis. The first and primary difference is this idea of synapsis. Remember, this happens in prophase 1. Synapsis is the process of homologous chromosomes pairing up, and it facilitates that crossing over thing, which increases genetic variability by creating recombinant genes, or genes that have genetic material from different sources. Another another primary difference between mitosis and meiosis is this idea of reduction division. From mitosis, we end up with two daughter cells that have the same number of chromosomes as the mother cell does. And presumably, all those chromosomes are genetically identical to those in the mother cell. However, in meiosis, we get a reduction division, where each daughter cell ends up with half the complement of chromosomes. And each of those chromosomes ought to be unique. Oops. So this is that same diagram, and I just kind of want to blast past it because I've drawn it right there. This is the same diagram that I drew over there, the difference between mitosis and meiosis. Okay, so we need to talk a little bit about sexual reproduction in the context of evolution, because this is its primary importance. We talked a little bit about the relative benefits, right, of sexual reproduction and asexual reproduction last time. Lots of the natural world still reproduces asexually because it's an incredibly effective and incredibly safe way to accomplish reproduction. The question is, if it's such a dangerous behavior, which it totally is, why do we do it? And the primary reason, in fact, probably the only reason that sexual reproduction is such a successful strategy is because of the introduction of genetic variation. So genetic variation will manifest itself at a variety of levels. Fundamentally, genetic variation just means differences between two different gene sequences. That's introduced by mutation that we'll talk about in the context of genetics next time. The way that sexual reproduction introduces genetic variability is as novel combinations of genes. We've talked about one way in which that occurs, right? That's crossing over. And what we get are recombinant chromosomes. Chromosomes that carry genetic material from two different sources. A chromosome of mom's that has some of dad's stuff on it. Secondly, there is this idea of an independent assortment of chromosomes. And this is an incredibly important one. We'll talk a lot more about it when we talk about genes or genetics. Is it okay if I erase this stuff over here? Okay, so when I initially drew our 4 chromosome cells,